Call out Eric Leire. I believe Eric is, or at least last I knew, he was in Belgium. He's somewhere in Europe. So thanks to all the European companies for staying up late for us. But is Eric online?
I'm here, and thank you for inviting me.
Hello, Eric. Please share your slides and go. We're looking forward to it.
I'm clicking my slides. So thank you very much for inviting me to this presentation, not only to present, but it's absolutely fantastic presentation. Let's jump to the presentation of Genflow. We usually define as a true longevity company. True longevity company means that we try to slow or reverse the aging biological processes. We want to do that both in human and in dogs. And to achieve this objective, we want to provide extra copies of sirtuin-6. We don't want to provide just a regular sirtuin-6 gene, but we want to provide a copy of sirtuin-6 genes that a variant of sirtuin-6 genes that found in centenarian, based on the work of Vera Gorbunova. We want also to deliver those genes, those extra genes, in a safe, ethical, efficient, and cost-effective manner. The purpose of my presentation is mostly to help us to close our seeding round of 1.5 million euros. We are based in Belgium, as Thomas mentioned, in the biopark that allow us to benefit from very generous financial support from the Wallonia region. The company is also defined by a fantastic team. I assume that the audience is very familiar with the people in my scientific advisory board. It's not only Vera Gorbunova that with a recent work on sirtuin-6 centenarian variant, but also Eric Verdin, Aubrey de Grey, Manlio Vinciguerra. In 10 minutes, I cannot really present everybody. On the board, we have also Andrew Scott, that's supposed to be very known among the audience, very good economist with books on longevity. Guy Alfano de Laurie is maybe less known. He was number 2 of Biogen. He's a veterinary and play a major role because vet is a big part of our strategy. The company is built on several beliefs. One of the beliefs is with the current demographics, we have a duty to consider aging as a risk factor that we need to treat. The second belief is that most of our aging is mostly controlled by our gene. I used to spend a lot of time explaining that mammals with different weight, same weight, have different lifespan, but Jeanne Calment with a glass of wine and a cigarette is making a better job than I do. One of the beliefs also is that we need to be acting as much as possible on the root cause of aging. As a longevity company, the first question you have is, what kind of longevity company are you? Are you a cellular senescence? Are you a mitochondrial company? We would like to be outside of those boxes. One of the beliefs we have is those 9 hallmarks of aging are interconnected. And what interconnect all those 9 hallmarks of aging is mostly the DNA damage and the lack of maintenance of DNA damage by epigenetic regulator genes that after a while become overworked. There's many of those genes, of course, who are responsible for the maintenance of aging. We try to shy away from big genes like FOXO or mTOR, and we focus on sirtuin-6. And to be more specific, on a variant of sirtuin-6, that's the mutation, there's 2 mutations, in fact, of the sirtuin-6 genes that are found in centenarian, and that's the genes we want to provide. We are also interested in sirtuin-6 because sirtuin-6 codes for 1 protein. So it's not like mTOR or even sirtuin-2 has multiple activities. But this protein has multiple activities, not only an histone deacetylase, but also through the ribosylation. It helps, for example, to upregulate PARP1. And therefore, it has an activity not only on DNA maintenance, but on shortener of telomere, on senescence. It's extremely important to act. We think it's extremely important to act upstream when you target to have an intervention on human and on dogs, on sophisticated living animals. In every lab, we can now extend the lifespan of C. elegans factor 10. It dropped to a factor 2 when we go to Drosophila. When it's on mice, it's only 25–50% depending. So extremely important to fit with the complexity of the aging problem as the complexity of the living organism is increasing with the sophistication. One of the criteria of choice of sirtuin-6 also is there's a huge literature of sirtuin-6, and also there's been a lot of animal models showing that adding extra copy of sirtuin-6 is benefit not only on the lifespan but the health span. The key question now is how do we deliver this sirtuin-6 gene? Of course, it has to be done ethically. Ethically means it has to be safe, it has to be cost effective, and it has to be efficient. The first biodistribution models we are running with mice are demonstrating that we can achieve an episomal expression of sirtuin-6 without any insertion, so in the DNA, so no risk of oncogenesis or mutagenesis. It's not a viral vector, so it's non-replicating, and our technology, our proprietary technology, allows us to have very limited immunogenicity, so we can repeat the injections, and we have a very interesting expression level of sirtuin-6 cross tissues through an IV infusion of 2 hours. The strategy we want to use is not something science fiction. It's a strategy that elephants are using to avoid cancer. Elephants are big animals, therefore they should have a lot of cancer. They don't. They don't. Why? Because they have extra copies of p53. What we want to do is to provide extra copies of sirtuin-6 gene and extra copy of a more efficient sirtuin-6 gene in repairing the double-strand break DNA, because it has a better ribosylation activity. So we expect that the extra copies of sirtuin-6 variant that we provide will translate into more DNA double-strand break repair, therefore an healthier longevity and maybe increasing the lifespan of the organism. The $1.5 million that we are looking for will allow us to have a 2 years runway and to achieve some two kind of objectives. One in dogs, to reach proof of concept in dogs that will allow us to sign a deal, a cooperation deal with one of the 4 players in veterinary. The second in human is to be able, after those 2 years, to open an IND that will allow us to start our clinical program. First, with a phase 1 in healthy volunteers. Second, with Werner syndrome. Werner syndrome is a rare progeria that allows us to have orphan drug status. It's also interesting because it's a progeria that happens with clusters. There's a Japanese cluster, not interesting for us, but there's a cluster in Italy, North Sardinia, that allows us to run a phase 2 clinical trial without too much challenging enrollment program. We will negotiate with several agencies or regulatory strategies to at least interact with as soon as possible with the European agency, the FDA with an interact meeting, the MHRA, the UK meeting, mostly discussing on the combination of surrogate marker. Right now, the cornerstone is GRIMAGE, but we think that will be a blend of multiple surrogate biomarkers will allow us to reach the our objective. In terms of competitive landscape, I think we have a competitive advantage versus a lot of companies because we are not boxed in one of the, we're not only solving 1/9 of the problem. Most of the company is very good at senescence, very good at mitochondria. We expect to have a competitive advantage by acting more upstream on multiple hallmark of aging. Also, our indication is clearly more close to pure aging indication. In 2 years, We'll reach a lot of key value milestones. We will reach not first in man, but first in dog. We'll have an IND that will allow us to start the clinical trial in Italy. And also that will allow us to have all the PD to be ready in terms of CMC, not only providing a good CMC section for IND, but to start the PD to have to be ready for large-scale commercial production, both for vet and for a clinical trial. We're raising 1.5 million. We have good traction with several types of VC, longevity specialized VCs and local seed VCs. We try also to expand our investor base to get more than 100 shareholders. It's a strategy I used in my previous company. I've been CEO of multiple biotech companies. The last one, I used OTCQB as an entry door to NASDAQ, where I list my company for $350 million market cap. This 1.5 million will allow us to reach enough milestones to be in good position to raise a 15 million round, A round. In conclusion, I will say that we are a disruptive company because we don't want to be boxed in only one hallmark. It's very ambitious, but we think we have risk mitigation with the veterinary program, which allow us to have a short-term potential collaboration, one of the 4 veterinary actors. We'll be able to have some very attractive milestone within 2 years, and if we succeed, we should have a significant bonus as first on market. I will emphasize also the very good, exceptional team behind this company. Thank you very much for giving us the opportunity to present.
Thank you so much, Eric. Jeff, do you want to unmute? And I don't know if you have any questions or observations. You're on mute.
Jeff, you're still on mute.
Jeff, can we hear you?
Mute. Okay, yeah, I'm sorry. I'm not able to unmute myself. Yeah, I thought that was an excellent presentation. The one question I had, you're raising 1.5 million euros, and you have a lot to do for that 1.5, including CMC manufacturing,
yes.
preparing an IND. I mean, you think 1.5 is really going to get you there?
The answer is the location in Belgium, in Charleroi, which is not the most appealing area in Belgium, but a lot of companies like Galapagos Bone started there. You benefit from a fantastic support from the Belgian government. 70% of my R&D is paid by the Wallonia, and in terms, it's in advance. It's a cash advance. It's not only a cash advance, it's a grant. So it gives me a lot of buffer in terms of financing. You're right.
Did you say 70%? 70? You said 70%?
Yes, 70. Yes. It's amazing.
Yeah.
Amazing.
So the 1.5 is really leveraging you another 2.5 million euros.
Exactly. It doubled my funding, and especially everything that's related to the manufacturing, all the CMC. It's 70% in cash advance. It's absolutely fantastic.
That is great. That's great. Yeah, you should highlight that here because
also,
that's a big advantage.
Close to the cluster of Werner syndrome, which is very important. If we were American, but within the 2 years, we'll have an American subsidiary because we will list the American subsidiary on OTCQB.
Super.
Great, excellent. Well, I'm sure the participants have questions for you. So perhaps those who are interested can go into a breakout room with Eric